Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be doing a final exam review. And keep in mind that these reviews are simply there to give you ideas for what to focus on or what to make sure that you know for the exam. They're not comprehensive reviews of the material, and they may or may not be comprehensive reviews of what's actually on the exam either. It's just a good foundation for what you need to know. And I'll be skipping through material. If I skip through material, it doesn't mean don't learn it, doesn't mean it won't appear on the exam, just means that I'm trying to highlight some of the meatier portions of the material that you definitely need to know. So with that disclaimer, let's go ahead and get started with chapter 10. Remember that this exam covers chapters 10 through 16 as well as chapter 18. The only chapter we're skipping there is 17. So here we've got transcription and RNA processing. Let's go ahead and get started. One thing I need you to know is the structure of RNA, a single strand molecule with a 3' end and a 5' end. And I need you to remember that RNA is more more unstable than DNA because of these 2' hydroxyl groups. They're very reactive. So what that means is RNA is more easily degraded than DNA, isn't it? And again, please know the structure of RNA and know that RNA can fold back in a complementary base pair with itself, right? You should know, obviously, that RNA is synthesized in a 5' to 3' fashion. RNA transcription goes in a 5' to 3' fashion. So here is the transcription start site. Do you remember that if this is a gene, the only transcription does not begin at the promoter. Transcription begins at the transcription start site, and it proceeds until the termination site. Do you remember sigma factor? Sigma factor allows the binding of the promoter with the RNA polymerase. What else? Do you remember these consensus sequences? This is the prokaryotic version of the Tata box, TATAAT, whereas in eukaryotes it's TATA, and that's the Pribnow box, and that's at -10 consensus. In general, you should know for the exam what a consensus sequence is. What is a consensus sequence? And then also know that the -35 consensus sequence, has a sequence as well, TTG-ACA. So here we're discussing consensus sequence. A consensus sequence consists of the most commonly encountered bases at each position in a group of related sequences, or a group of related organisms, or between different organisms. It's a consensus. What else? Obviously, know the process of transcription. We spent some time talking about how transcription is initiated. This is initiation of transcription, elongation of transcription, and finally, termination of transcription can be row dependent or row independent. What else? Definitely know the concept of intron splicing. You splice the introns out, and remember, there is no RNA processing that occurs in prokaryotes. Only eukaryotes undergo RNA processing by means of 5' cap, methylguanine cap, 3' poly A tail, and removal or excision of the introns. You excise the introns. Okay, you should know what the Shine-Dalgarno sequence is. And remember, again, the 3 pre-mRNA or modifications that occur in eukaryotes: the 5' cap, the 3' poly A tail, and excision of the introns. Oh yes, you should know how the consensus sequence works for intron removal. So you've got the 5' consensus sequence, the 3' consensus sequence, followed by a branch point in the middle. And this facilitates in the spliceosome, remember the spliceosome enzyme, removing the introns. Also here, this is very important to know for the exam. Remember that this is the consensus sequence required for marking the cleavage site for the poly A tail. Remember, on eukaryotic pre-mRNA, when you see this consensus site, AAUAAA, that marks the cleavage site 11 to 30 nucleotides away, the cleavage site for polyadenylation to occur, where the poly A tail occurs. So please remember to know that for the exam. And here are the consensus sequences for the intron splicing. You have the 5' consensus sequence we talked about, the 3' consensus sequence we talked about, as well as a branch point A. Okay. Now, here's another very important point to know for the exam, and it's this idea of alternative splicing. So you should know the concept of alternative splicing. And what you should know is that it is a way of getting different mRNA from the same pre-mRNA and from the same gene, right? So the same gene gives you different proteins. How? By splicing the pre-mRNA different ways. Does that make sense? All right, and this is just a rundown of all of the different modifications. You've got the 5' cap, you've got your 3' poly A tail after the cleavage site, consensus sequence AAUAAA, and you have RNA splicing. And remember that RNA splicing of the pre-mRNA to mRNA can occur in different ways, and that's called alternative splicing. Obviously, remember that eukaryotes have introns and exons. Prokaryotes do not have introns. They only have one big exon. Please remember that prokaryotes have operon. Prokaryotes have operon and operators on their operon, whereas eukaryotes do not have operon. They don't have technically operators. Instead, eukaryotes have transcription factor binding sites. tRNA, you should know the role of tRNA is to bind different amino acids. The tRNA get charged on their 3' end with amino acids. Let's see what else we should touch on. Yeah. No, we're doing good. We're doing well on this review. Again, so you've got your tRNA, and remember, you see here on the tRNA you have your acceptor arm. The acceptor arm is the 3' end of the tRNA molecule, and that's called the acceptor arm because it's going to accept a amino acid. It's going to be charged with an amino acid. Oh, this is very important too for you to understand how microRNAs and small interfering RNAs work. So make sure you understand how microRNAs and small interfering RNAs work. And remember, they're cleaved by dicer. They're cleaved by dicer. Double-strand RNA is cleaved by dicer. And then the small RNAs pair up with the RNA-induced silencing complex RISC, which then base pairs with the mRNA that you want to silence, and you either cleave the mRNA, small interfering RNAs cleave and degrade the mRNA, the target mRNA, whereas microRNAs bind to and just gum up the works. It inhibits translation. We should also know how CRISPR works. You have CRISPR. Your cRNA pairs up with Cas protein to form an effector complex. This effector complex binds to foreign DNA and promotes cleaving of the foreign DNA. Awesome. So that about covers chapter 10. Let's move on. Let's move on to chapter 11. What do you say? Let me find chapter 11 here. Okay. All right, chapter 11. Now we're talking about the second part of gene expression. We're talking about translation. Translation. What do we need to know about translation? Remember that there are 20 naturally occurring amino acids. You should know the structure of an amino acid. You have the alpha carbon, you have an amino group, a carboxyl group, a hydrogen, and this all is called the backbone of the amino acid. This all is called the backbone because all the amino acids have this backbone of 1, 2, 3. And where the amino acids differ is their number 4, this radical group. It's also called the R side chain. It's also just called a side chain. Or, sometimes it's even called the variable side chain. Remember that amino acids are linked together by peptide bonds. And do you remember what's responsible for making the peptide bonds? That's right, it's the P site of the ribosome. That P site is responsible for peptidyltransferase activity. Also remember, every protein has an amino end called the N terminus, and every protein has a carboxyl end or a C terminus. Here's something you need to know about protein structure for the exam. What's responsible for the secondary structure? The alpha helices and beta sheets that make up the secondary structure of a protein. The thing responsible for that is the amino acid sequence, or the primary structure. Does that make sense? The amino acid sequence of a protein, also known as the primary structure of a protein, is responsible for the secondary and the tertiary structures of that protein. So definitely know that for the exam. The genetic code, we talk a lot about the genetic code. Remember, there are 64 different codons. 3 of them code for stop. Remember, TAA, TGA, TAG, those 3. Or instead of T, you could say U, UAA, UGA, UAG. Those are the stop codons. They do not code for any kind of amino acid, do they? Remember that it's release factor. It's a protein called release factor that recognizes the stop codons, not a tRNA. A tRNA does not recognize the stop codons. tRNA recognize the other 61 codons. tRNA recognize the other 61 codons because the other 61 codons code for amino acids. But there's no tRNA that recognizes the stop codons. Instead, a protein called release factor recognizes the stop codons. You should know what a codon is. 3 nucleotides that encode an amino acid. You should know that the genetic code is degenerate. You have synonymous codons, also known as redundant codons. Okay, and you should know, again, the stop codons, UAA, UAG, UGA, and the start codon is AUG. The start codon is AUG, and it codes for what amino acid? Methionine. Methionine. Okay, do you remember why you have multiple different codons coding for the same thing? So why does CUU, CUC, CUA, CUG, why do they all code for leucine? Remember, it's because the genetic code is redundant or degenerate. The genetic code is redundant or degenerate, which means that multiple different codons can code for the same thing. And remember what causes that? It was wobble base pairing, right? The wobble base pairing, which is shown here. The third position on the codon forms a weak connection with the tRNA anticodon. And because of that, the same tRNA might be able to bind to more than one codon. So a single tRNA can bind with more than one codon. So another way of saying that, remember, because the anticodon are on the tRNA, so a single anticodon can pair with more than one codon. Another way of saying that is a single tRNA can bind to more than one codon. So D. Okay. Reading frame, you should know what the reading frame is, and it's set by what? It's set by the start codon. In which direction does translation proceed? The ribosome proceeds from the 5' near the 5' end of the mRNA. It proceeds towards the 3' end of the mRNA. Okay. You should know about initiation, elongation, and termination of translation. So for example, you should know that during initiation, for the exam, you should know that during initiation, this requires the initiator tRNA, which is attached to N-formal methionine. So that's F-met tRNA. You also need GTP. You also need GTP, and you also need the initiation factor 3. So you should know that those are the factors you need for initiating translation. Again, you should know what the Shine-Dalgarno sequence is versus the Kozak sequence. Remember, one's found in prokaryotes and one's found in eukaryotes. Again, for the exam, know how this initiation works. You have the small subunit of the ribosome. It finds the AUG start codon. Initiation factor 3 binds. F-met binds. So your start tRNA, called tRNA with N-formal methionine, binds with GTP, IF2, and IF1, right? All of these factors are responsible during for initiation of translation. Remember, the large subunit is not, attached yet at this point. Only after all these other factors attach does the large subunit attach. Okay. So do you know what would happen if you were to cleave the tail here of the mRNA? If you were to cleave the mRNA tail, that would lead to degradation of the mRNA, wouldn't it? If you cleave the mRNA here somewhere, that would lead to degradation of the mRNA. It's because this poly A tail loops back and forms a stabilizing structure here with the cap, the 5' cap proteins. You should know this. These are the sites of the ribosome. The E site of the ribosome, that's where uncharged tRNA exit. The P site, remember, that's where peptidyltransferase activity occurs. That's where the peptide bonds are made. And then you've got the A site. I call it the accepting site. Your books, your book makes sure to call it the aminoacyl site. But this is where you should know for the exam, you should know that the A site is where new charged tRNA come in to bind to codons, right? So this is the process of elongation. So be familiar with this process of elongation. Again, charged tRNA come into the A site. The P site makes the peptide bond with its peptidyltransferase activity, and the E site ejects the uncharged tRNA after it's done its job. This is the process of elongation again. Again, do you know what release factors are? These are proteins that recognize the stop codons. And they induce what? They induce termination of translation. They dissociate the ribosome from the mRNA and all the tRNA. Great. You should know what polyribosomes are. You should definitely know this. Polyribosomes, an mRNA with several ribosomes attached, can occur in both prokaryotes and eukaryotes. So please know that. Okay, this is the polyribosome in action. And then that is about it. Let me double check and make sure we touched on everything. Yeah. Yep. Again, like I mentioned, know all of the players that play a role during initiation of translation, elongation of translation, and termination of translation. What are all the players that play a role? What are all the important parts to each of those processes? Okay. Okay, and do you remember when you link amino acids together? What is that called? First when you link amino acids together, you form a peptide, right? A polypeptide. And then eventually, when you're done translation, that long polypeptide is called a protein. Does that make sense? So remember that for the exam. Great. Yeah, I believe we touched on everything in this chapter. Great. So let's move on to the next chapter. Chapter 12. All right. We're getting there. All right, so chapter 12, control of gene expression. Let's talk about control of gene expression. You should know these definitions, obviously: structural genes, regulatory elements. And we made a big deal about positive control and negative control. So let me bring that up in a minute. Oh, okay. Before we talk about that, operon. Do eukaryotes have operon? No, only seen in prokaryotic cells. Operon and operators, and remember the operator is part of the operon, these are found in prokaryotes only, not in eukaryotes. Okay, so here you have an example of an operon. Please remember how the operon works. You've got a separate regulator gene. You've got the operon itself with its operator, promoter, gene A, B, and C, for instance. Please know what the operator's job is, what the promoter's job is. Remember that the regulator protein can either be a repressor or some kind of activator. And here's the important stuff to know as well. What is negative control of a operon? You've got the repressor. The regulatory protein is a repressor. What's positive control? The regulatory protein is an activator. What are inducible operons? These are ones that are normally off, and something has to be done to turn them on. What are repressible operons? These are normally on, and something has to be done to turn them off. Again, please know these examples. What's going on in a negative inducible, a positive inducible, negative repressible, and positive repressible system, operon system? How do they function? The lac operon. Please know the lac operon in detail. What does lac I code for? Lac P, lac O. What is it involved in? Lactose metabolism. What's the inducer? Allolactose. What are the structural genes of the operon? Lac Z codes for beta-galactosidase, lac Y for permease, and lac A transacetylase. All right, and how does it function? Remember, beta-galactosidase breaks down lactose into galactose and glucose. It also converts lactose to allolactose, which is the inducer. And permease allows for transport of lactose into the cell. Okay, and please understand how this system works. There may be some questions about the lac operon. So for instance, is it, what type of system is it? So you've got a repressor, right? You've got a repressor, and the repressor binds to the operator. So you should know what would happen if the repressor couldn't bind to the operator, or what would happen if the repressor stayed on the operator. Okay. What else? Again, know how this system works, right? And again, just to explain it one more time, your regulator gene encodes for a repressor. This repressor is active all the time in the absence of allolactose. So this repressor is an active repressor. The repressor binds to the LAC-O operator, preventing RNA polymerase from transcribing these genes. However, in the presence of allolactose, the repressor becomes inactive, allowing for RNA polymerase to attach to the promoter and promote gene transcription. Okay. Histone structure and modification. Remember, you could methylate histone tails. This causes promoters to be switched off. You can acetylate histone tails. This reveals the chromatin structure, allowing transcription to carry out. Remember, you should know about the basal transcription apparatus and transcriptional regulator proteins. So remember that the basal transcription apparatus, this is your general transcription factors as well as RNA polymerase, and it's capable of transcribing genes to minimal levels. Minimal levels. All right, but with the help of transcriptional regulator proteins, these allow you to transcribe to normal levels. They help the basal transcription apparatus to transcribe to normal higher levels. Okay, so definitely know what transcriptional regulator proteins are doing. Know the term enhancers, insulator, know the term insulator. So no enhancer, no insulator. Again, know how RNAi works. RNAi stands for RNA inhibition, right? So the double-stranded RNA is broken down to siRNA. Know how this process works. Again, dicer cleaves double-strand RNA into small RNAs, siRNAs, which are known as small interfering RNAs. These small interfering RNAs then combine with the RISC complex, and then together they bind to the target mRNA and mark it for cleavage, right? Cleavage. So there's a cleavage event here that leads to degradation of the target mRNA. Okay, but with microRNAs, you have a hairpin loop here, and dicer cleaves that into double-strand, into small RNAs, I should say, microRNAs, which complex with the RISC complex. They seek out target mRNAs, and in this case, they don't degrade the target mRNA, they inhibit translation. Okay, and don't forget that also siRNAs, in addition to destroying the target mRNA through cleavage, they can also associate with the DNA and mark it for methylation. Remember that you can methylate DNA, so please know about methylation of DNA, and know that you could methylate DNA at cytosine residues. Cytosine is methylated. These are your CpG dinucleotides. That's where the methylation occurs, at cytosine. Okay, let's see if we've skipped anything. All right, next chapter. So I think we touched on most of the important things there. Before we hop into the next chapter, let's take a quick break time with Gizmo and Wicket, our final break time with Gizmo and Wicket for the semester, and we'll be right back to finish this final exam review. All right, welcome back from break time with Gizmo and Wicket. Let's keep going with our final exam review with chapter 13. We're touching on mutations in this chapter. You should know about somatic mutations, germline mutations. One thing I want you to remember is that only germline mutations are passed on to your offspring, not somatic mutations. What else? You should know what a transition base substitution mutation is versus a transversion. Remember, what are the purines? What are the pyrimidines? So know the difference between purines, pyrimidines. What is an example of a transition? What is an example of a transversion? Insertions and deletions. You should know that frame shift mutations are really bad. Probably the worst type of mutation you could get, frame shift mutations. Especially in terms of point mutations. Frame shifts are bad. You should know that. Now, if you have insertion of one nucleotide or deletion of one nucleotide, that could lead to a frame shift. But remember that if you have insertion or deletion of multiples of 3 nucleotides, that's not a frame shift, because if you remove 3 nucleotides, you've removed one whole codon. You haven't actually shifted the reading frame. Just remember that as well. Understand that concept. You should know what base substitution, base insertion, base deletion mutations are. Again, I told you you should understand this concept in detail of transversions versus transitions. All right, you should know what is forward mutation. For the exam, you should know forward mutation is wild type. You start with a wild type, and then you end up with a mutant. What's a reverse mutation? A mutant reverts back to wild type. What's a missense mutation? You should know this. A different amino acid, right? The codon is mutated in such a way that you end up with a coding for a different amino acid. Nonsense mutation. You code for a stop codon, right? A stop codon. So a sense codon becomes a nonsense codon, which means a stop codon, right? A stop codon. Silent mutation, you should know that this is a codon that gets mutated. However, there's no change in the protein because it still codes for the same amino acid. What else? Oh, you should know what is a conditional mutation and a lethal mutation. Again, I told you missense, nonsense, silent. Please know those types of mutations. Spontaneous mutations versus induced mutations. Know that concept there. The causes, you should know the causes of spontaneous mutations. You should know the causes of that. And mutagen, you should definitely know what is a mutagen, the definition of mutagen, what is a mutagen. Base analogs, you should know this. Base analogs, a class of chemical mutagens with structures similar to those of the 4 standard bases. So remember, the base analogs, because they're so similar to the nucleotides, they get incorporated into the DNA, causing mutation. I believe, just know these terms: alkylating agents, deamination, hydroxylamine. These are chemically induced mutations. Intercalating agents, how do they work? Oh, you should definitely know this. What is the problem with UV radiation? Remember, UV radiation causes pyrimidine dimers, like thymine dimers, or pyrimidine dimers in general, like cytosine dimers, or cytosine thymine dimers, and that could lead to mutation, right? You should know this for the exam. It's a big, important point, that DNA polymerase has proofreading ability. DNA polymerase has proofreading ability. Does RNA polymerase have proofreading ability? No. And remember, I mentioned this before. RNA viruses, such as the virus for COVID, SARS-CoV-2, and the virus for the flu, the flu virus, these are RNA viruses. And their genomes are copied by an RNA polymerase. RNA polymerase doesn't have this proofreading 3 to 5 prime exonuclease activity. It can't check its work. And this is why you're always ending up with mutations of the flu virus. You're getting new strains of the flu virus, and you're always ending up with new strains of COVID, new strains of SARS-CoV-2. This is why they're so concerned about new strains of SARS-CoV-2 popping up around the world. Yeah, I just heard about a new strain today. The reason is, if you're not checking your work, you make mistakes. DNA polymerase can check its work, it has proofreading. RNA polymerase can't check its work. Mismatch repair. You should know about some of these repair mechanisms. So how does this mismatch repair complex work, right? So remember, it looks for this CTAG methyl group, It looks for this methyl group on the old strand, the template strand, and it uses that old strand as a template for synthesis of a new strand, of a daughter strand, to repair the DNA, to repair the mismatch. So you should know how this functions. All right, so that leads us to the end of that chapter. Let me take a quick look here and make sure we touched on most everything. Okay, one thing we didn't touch on directly, but indirectly, because if you remember, we skipped a lot of the material on transposable elements, but you know what transposable elements are from a previous chapter. Why would a transposable element meaning a piece of DNA that could remove itself from one part of the genome and insert itself in another part of a genome, how would a transposable element cause a mutation, do you think? Right? So if a piece of DNA can remove itself from one part of the genome and insert itself into another part of your genome, couldn't it maybe disrupt a gene? Couldn't it insert itself in the middle of an exon, for instance? And wouldn't that disrupt your gene? So think about that for the exam. Like, how would a transposable element cause DNA damage? Well, perhaps it could insert itself into a gene, right? And then disrupt the gene function. Okay, with that, let's hop to the next chapter. Chapter 14. Let's hop right into it. Recombinant DNA technology. Remember, the techniques include locating, isolating, altering, and studying DNA segments. You should know what restriction enzymes are. Definitely know what these restriction endonucleases are. And you should know that they can leave either cohesive ends or they could leave blunt ends, right? They could leave cohesive ends or blunt ends. And do you remember where they come from? They come from bacteria. They are naturally occurring in bacteria, and we use them in the molecular biology lab. You should know the role of ligase. Ligase's job is to attach these ends together by sealing the phosphodiester backbone bonds. CRISPR-Cas9, we talked about CRISPR and Cas9. Please understand how this system functions. It's very important to understand. You have your sgRNA, which includes your crRNA, tracer RNA, that complexes with the Cas9 protein to form the effector complex. The effector complex locates the PAM sequence on the target DNA. Then the sgDNA can bind to the target DNA, inducing cleavage. Cas9 will cleave the target DNA. And remember that if non-homologous end joining were to occur at that point, this would cause a deletion or duplication, and you could destroy genes this way. You could turn off genes this way. However, if you provide a donor DNA through homologous recombination, if that's the outcome, you could actually insert DNA. So you could fix a gene, for instance. So this is really super cool. If you guys can understand this, if you're going to go into medicine, there's a lot of talk about CRISPR and how it works. Just understand that you could target any sequence you want in the genome. You could target it, right? So let's say a gene that you want to mess with, you could target that with your sgRNA. You could target that sequence. Then you could go in and you can either cause destruction of that gene, so you turn that gene off, completely, or cause the protein not to form, or you can try to repair that gene and fix a problem, right? So isn't that cool? Gel electrophoresis. Please know how this works. Remember that two things. One is the DNA, because DNA is negatively charged, the DNA will run towards the positive charge in the gel, right? So the DNA goes towards the cathode, towards the positive end. Also, you should know that larger pieces or larger fragments of DNA travel slower than smaller fragments of DNA. You should know this too. For the exam, I want you to know this. If you want to detect a specific gene or a specific sequence of DNA, you're going to do southern blotting with a DNA probe. So southern blotting detects specific DNA sequences. Northern blotting, you would do this technique to detect RNA, specific RNA sequences. Western blotting, you would do this technique in the lab to detect specific proteins, and you use special antibodies, specific antibodies towards those proteins to identify those proteins. Did you follow that? You want to detect specific DNA, southern blot, specific RNA, northern blot, specific protein, western blot. Know that for the exam. PCR, remember polymerase chain reaction? I want you to know about this. What is PCR and what is Taq polymerase? Why would you need Taq polymerase? This is the DNA polymerase. This is what's going to copy the DNA. And remember that you're going to need certain things for PCR, right? You need primers. These are your oligonucleotide primers. You're going to need your Taq polymerase. You're going to need the template DNA. All right, let me just make sure we're touching on some of these important things. And remember, what is the purpose of PCR? Is to amplify DNA, right? So you start with a little bit of DNA, and then with PCR, you could magnify that DNA. You could multiply it into many copies of that stretch of DNA, correct? Okay. So in gene cloning, we need to understand cloning. So please know what a plasmid is. What is transformation? You should know for the exam that there are different kinds of gene vectors during cloning, right? You have plasmids. You need to know that you have plasmids. You have cosmids. And then you can have the artificial chromosomes, like bacterial artificial chromosome and yeast artificial chromosome. All of these are vectors, expression vectors, or cloning vectors, they call them. Okay, let me make sure we're touching on everything. All right. All right, so I want you to understand the process of cloning, right? So you digest your plasmid, you digest your foreign DNA, your insert DNA. You then allow for complementary base pairing with the sticky ends. Then you do ligation, right? So you ligate into the vector. You ligate into the vector. You then transform the recipient cells, right? And then you can identify the transformants. Remember, so cloning a gene after isolating it requires what actions? So you clone a gene, you then transform bacteria, and then you screen for recombinants. Remember that we can use microsatellites in order to make DNA profiles and to link individuals to crime scene samples. Okay, forward and reverse genetics. So you should know what forward genetics means. Actually, you should know forward genetics. You begin with a phenotype, and then you begin with the phenotype, and then you figure out what the gene is responsible for that phenotype. Reverse genetics, you begin with a gene of interest, and then and you have no idea what that gene does, and then you trace that to a phenotype. Does that make sense? Please know the difference between forward genetics and reverse genetics. A transgenic mouse. What is a knockout mouse, a knock-in mouse? That's great. I think we have touched on everything from here. All right, here we are in chapter 15. Let's go ahead and get started. You should know what structural genomics is. What is a genetic map? You should know what a genetic map is and the limitations of the genetic map. You should know what is a physical map. Know all of the ins and outs of a physical map, all of the ins and outs of a genetic map. I want you to be able to compare and contrast the two. You should know about the sequencing project. What is the difference between the map-based sequencing project and the whole genome shotgun sequencing? What was the difference there? What happened? Which one included the use of a contig, right, map-based? And the whole genome shotgun, how was that different than map-based sequencing? Remember with the whole genome, we just took fragments and we then sequenced the fragments of DNA, and then using computer algorithms, we were able to assemble the entire sequence back together. You should know what a contig is. So again, know what a genetic map is, a physical map. Which one's more accurate? Which one has higher resolution, a physical map or a genetic map? Know about the shotgun approach. All right, single nucleotide polymorphisms. Know what they are and why we study them, right? What are single nucleotide polymorphisms? Very, very important stuff. Okay, what is a haplotype? What is a haplotype? Oh, what is synthetic biology? You're creating chromosomes or organisms from scratch. What's functional genomics? What is the transcriptome versus the proteome? Know the term proteome. What do you study with microarrays? Remember, with microarrays, you can study gene expression, right? Okay, compare and contrast prokaryotic genomes with eukaryotic genomes. Remember, with prokaryotic genomes, they're smaller genome size, but there is a positive correlation with gene number and size of the genome, right? What else? So remember, because there is a positive correlation between genes number and genome size, there's a higher density of genes, right? There's a higher gene density in the genome, isn't there? Whereas with eukaryotes, remember the genome size is huge, but the genome size doesn't really correlate with the organism complexity. The genome size doesn't even correlate with the number of genes present, right? So you're going to have less gene density. You're going to have less gene density. Okay, very good. We talked about transposable elements a little bit. Remember, transposable elements, if they insert themselves into a gene, they could disrupt that gene. And transposable elements have made up to 45% of the human genome. Now they don't work anymore. They can't transpose themselves. They're defective. However, they make up 45% of our genome, which is fascinating. Remember that only eukaryotes have exons and introns, right? And not much of our actual DNA encodes for protein. Only about 1.5% of our DNA encodes protein. What else do we need to know? Again, know what the proteome is. What's proteomics? Great. I think we touched on everything there. So let's go ahead and hop into the next chapter. All right, this is a neat chapter. I really enjoyed this chapter. You should know how cancers cause problems, right? Remember this, that cancer cells crowd out normal cells. Like you see here, the cancer cells in the liver, the breast cancer cells in the liver are crowding out the liver cells. And not only that, but they're robbing the healthy cells of nutrients. So this is why cancer kills. So you should know that. You should know how tumor formation occurs and what is a tumor, the difference between benign, malignant tumors and metastasis. Clonal evolution. You should know this term, clonal evolution. Tumor cells acquire more mutations over time that allow them to become increasingly more aggressive. So know this concept of clonal evolution. Know the concept of clonal evolution for the exam. What does the concept mean? How does it work? You should know that there are these contributing factors to cancer. Obviously, environmental factors can cause cancer, but also spontaneous factors can cause cancer. You should know the definitions of oncogene, proto-oncogene, and tumor suppressor gene. Definitely know the difference between proto-oncogenes and oncogenes. What's the difference between the two? Okay, remember oncogenes are dominant acting, tumor suppressor genes are recessive acting. Oncogenes promote cell division and cell proliferation, whereas tumor suppressor genes stop cell division. They stop the cell from dividing when it's not supposed to. Proto-oncogenes, remember that these are your normal everyday genes that when misregulated or mutated can cause cancer, can become oncogenes. Okay. Remember this, cancer cells, they do not respond to normal cell cycle controls, right? Does that make sense? Remember that cancer cells, they've lost the ability to respond to normal cell cycle control, like proliferation controls or, controls that are telling the cell to stop proliferating. All right, what else? Okay. Telomerase. You should know the importance of telomerase, right? Overexpression of telomerase is found in cancer cells. Why? Because every time your DNA is copied and the cell divides, your chromosomes shorten in length, and that causes cells to not be able to divide too many times, right? But when telomerase is active in somatic cells, where it normally is not active, the ends of your chromosomes do not shorten with each cell division, and the cells effectively become immortalized, which means that the cells can divide and divide and divide. Does that make sense? So in cancer cells, you're going to see too much telomerase activity, not too little. Angiogenesis, remember that blood vessel stimulation or growth of new blood vessels is important for tumors to grow past a certain size, right? Remember, the tumors send out signals like little hormones, like for example VEGF, vascular endothelial growth factor, and that causes blood vessels to grow into the tumor, right, and allow the tumor to have its own blood supply, so the tumor can get really big, right? But if you don't allow blood vessels to grow into the tumor and you block angiogenesis, well, then the tumor is not going to get past a certain size. Isn't that interesting? So we should note that for the exam. Okay. What else? tumor progression model, please understand the tumor progression model. There's no one gene that really causes cancer usually. Usually it's a series of mutations that cause cancer. Remember, you frequently find aneuploidy in cancer. You frequently find chromosomal mutations in cancer. You should know how a retrovirus may cause cancer, right? You should know how a retrovirus may cause cancer. So, for example, do you remember these two? There's two ways. A retrovirus might mutate or rearrange a proto-oncogene to cause cancer, or a retrovirus might insert a strong promoter near one of your proto-oncogenes to cause cancer. And that's a problem because, remember, misregulation of proto-oncogenes can cause cancer. Awesome. I think that about wraps up that chapter. Let's hop to the last chapter. What do you say? All right, time for the last chapter, chapter 18. There isn't a whole lot in this chapter. You know, I limited this chapter a little bit, but I still kept some of the very important points. So let's get started with this. You should know that genetic variation is very important in populations for their fitness, for their ability to evolve and to continue to exist, right? You limit gene variation, that's a good way to go extinct. You should know what genetic drift is. What is the definition of evolution? The frequency, what does that mean? Frequency, allelic frequency versus genic frequencies. Now, Hardy-Weinberg law, we should definitely know the Hardy-Weinberg law with Hardy-Weinberg equilibrium. I want you to know the assumptions here. For Hardy-Weinberg equilibrium to form, to hold up, you need randomly mating population. Most importantly, you need a large population. You need no effect of mutation, migration, and you need no natural selection. So these are the stipulations for the Hardy-Weinberg law, the assumptions. And then, you know, the predictions are the allelic frequencies of a population won't change, and the genic frequencies stabilize. So definitely know that. What else? Now, okay, you should know what is the consequence of small population size. What happens with small population size? And remember, you can have genetic drift, right? You can have genetic drift. And what are the effects of genetic drift? You should know that when you have small population size, the consequence is genetic drift, and the effects of genetic drift are 1, 2, and 3. You could get fixation of different alleles in the population, right? So that's very, very important to know. You should know the definition of natural selection. What is selective agents? What does fitness mean? Okay, and that really wraps up this whole final exam. I hope this was a good review. It's been a pleasure to be an instructor for this semester. I think that about wraps up everything. I hope you enjoyed learning the material. I've had a passion for genetics since I was a youngster. In 9th grade, I think it was in 9th grade, I decided I want to learn genetics. So I went out and I just bought a genetics book and read it wherever I could. So I've had a passion for this stuff for most of my life now, and I got my PhD. I ended up getting my PhD in genetics and doing postdoctoral research in genetics. So I actually did end up following my dream and learning this material and sharing it with you. So I hope I was able to share it in a good way. I love sharing my passion, and I hope you took away some cool and interesting material about genetics and how important genetics is to the fields of medicine and research as well. And you know, as most of you are going on to become doctors, nurses, you're going on to become pharmacists, etc. You know, a strong understanding of genetics is going to help you in your careers, and I hope you took away a lot from it. I hope Gizmo and Wicket were fun for you to watch, and you know, good co-hosts of the show as well. And I hope you have a great break after this semester. And again, it was a pleasure. I hope you learned a lot, and leave any comments or questions in the box below, and I'll catch you guys next time.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D.